Okay, so this is a map of the United States highlighting the obesity rates per state. And as you can see, most of the nation has between 30 and 30% obesity rate. And if you look at Europe, they don't have a single country over 30%, and they have a few actually under 20. So this has become a huge problem in the United States, with 36.5% of our adult population categorized as obese. And a third of the deaths in the United States can be attributed to cardiovascular diseases, which are often linked to obesity. And obesity is unique in the sense that you can't really take a pill and be cured. There's no real cure for this, but there are some, obviously, there's therapies. You have your regular diet with exercise, and then they do surgeries now, lap bands, they basically constrict your stomach size so you can't eat as much. The gastric sleeve, they cut out a piece of your stomach. But this paper is going to focus on calorie restriction mostly. My paper is called Calorie Restriction Leads to Browning of White Adipose Tissue Through Type 2 Immune Signaling, or in layman's terms, Why Low-Calorie Diets Result in Weight Loss. So to start things off, I just define obesity. It's a condition characterized by the excessive storage and accumulation of fat in the body. People who are obese have a BMI of 30 to 40, and extremely obese is over 40. And BMI is just kilograms over meters squared, so height. So can anybody tell me why the excessive accumulation of fat in the body would be a problem?
Interferes with blood vessels?
Yeah. Okay. So some of the physiological problems with obesity, high blood glucose levels and the inability to process that blood glucose, and we all know that as diabetes. And then muscles are inefficient in their oxygen consumption, so they have a harder time taking up oxygen and like just using it. And that just gets in the way of basically everything, like walking upstairs and just regular body functions. And what this paper is going to really key in on is something called brown tissue, and the difference between white and brown tissue. So most obese people have white fat, and we want to have brown tissue. So white fat is basically less dense, it takes up a lot more space, and it puts your organs and your body under a lot more pressure, whereas brown tissue is denser and organs are under a lot less stress, and your body is just under less stress in general. So important vocabulary before I really jump into the paper. Type 2 signaling is an immune response pathway. Type 2 cytokines are the proteins associated with this pathway. And then two regions that we're going to look at are the ING-SAT and the PG-VAT. The ING-SAT is the region of fat directly under the skin, whereas the PG-VAT is visceral fat, and that is surrounding the organs and farther under the skin. So the experimental design, they had two groups. They had an AL group, which had a regular diet, and then they had a calorie-restricted group, and they restricted the calories by 40%. So they were eating 60% of what the AL group was. And the first thing that they looked at was the browning of white tissue. And that white tissue is what a lot of obese people have, and we want to have brown tissue, which is a lot better. So in figure 8 here, they were looking at proteins that were involved in brown tissue, in the formation of brown tissue, and they already knew what these proteins were. And as you can see, in every single one of these proteins, CR mice had a significant upregulation as opposed to the regular diet mice. And that just shows us that there was a lot more of the conversion of white tissue to brown tissue in the calorie-restricted mice. And in figure E here, they looked at the ING-SAT, they looked at actual tissue from that region of fat right under the skin. And they basically, just as a visual representation of the browning of the tissue in the AL group, you can see that the cells aren't really densely packed together. And then in the CR group, you can tell that they're much more densely packed together, indicating the presence of brown tissue. So after they confirmed the presence of this brown tissue, they wanted to look at the benefits of it. Like, why is this good? Okay.
How long were the mice on the diets for? Did it say?
I'm not sure. So then they looked at the benefits of this brown tissue. Now that we've established that these CR mice have a lot more of it, why is that a good thing? So they looked at two things. They looked at oxygen consumption rate, which is OCR, and they looked at blood glucose levels. So in figure B here, they're looking at the OCR, and the calorie-restricted mice were able to absorb oxygen in their tissue twice as fast as the group with a regular diet. And that's important because one of the big problems with obesity is that people that are obese have a difficulty taking up oxygen efficiently. And then in figure E, they were looking at blood glucose levels. They injected them with sugar, both groups with sugar, and they tracked at 30-minute intervals for 120 minutes the blood glucose levels. And these are OB mice, so that means that they are bound to be obese, and they have the obesity gene, and they're going to be obese. There's nothing they can do about it. And in the regular group, from the time after the injection to 120 minutes, the blood glucose levels did not change. They were not able to process it, and the levels did not lower. However, the calorie-restricted mice, in that time frame from 0 to 120 minutes, their blood glucose levels were cut in half, and they were able to process it much more efficiently. And this is a big deal because obese people, they generally, they could have obesity. Diabetes. They're more prone to diabetes because they can't process blood glucose better. And the CR mice were able to do this very well. So why would these be beneficial to obese people, these metabolic effects? Dr. Kirk.
I was actually going to ask a question. Do you know the experiment E there? Did they also do that in wild type mice? And
I don't think so. They just did it with. So why would this be beneficial to obese people?
For the blood sugar levels, like where are you supposed to be? Like for the blood glucose,
yeah.
What's like the ideal number you're looking for there?
Well, I think we're just looking for that number to go down after 120 minutes. Not necessarily go to 0, but like significantly go down. But the regular group, it kind of went up a little bit. Like it just stayed the same. They just wanted to see that the calorie-restricted mice, their levels went down much more than the regular diet. So why would this be beneficial?
Well, if you're more efficient with your oxygen, everyday tasks are easier, and you have better blood sugar, right? Is that what I'm talking about? Better blood sugar, then you're not basically it's not just healthier. If you have a higher blood sugar, you're at risk for diabetes and other things that put you at risk for.
Yeah, absolutely. So now we're going to start creating a pathway. So we know that calorie restriction leads to the browning of white tissue, and that brown tissue has its metabolic effects, such as oxygen consumption rate, glucose levels, and then we know that there's a weight loss associated with the calorie restriction. That's been established. But the scientists thought that there was a missing link in between the calorie restriction and the brown tissue. They wanted to find out what this whole pathway was dependent upon, what the whole mechanism was dependent upon. And they looked at type 2 cytokines. Type 2 cytokines are the proteins involved in the type 2 immune signaling pathway, so they're involved with the immune response. And they found that in calorie-restricted mice, they had a much higher levels of these proteins. As you can see in A, they looked at 3 of them, IL-4, IL-5, and IL-13. They're all type 2 cytokines, and they were all significantly upregulated in the calorie-restricted mice. And in that, they looked at eosinophils in the ING sat, which is that region of fat right under the skin. And eosinophils, they are immune cells associated with the type 2 immune signaling pathway. And they found that the CR mice had double the amount of these cells in the ING sat as opposed to the regular diet mice. So once they established that there was more type 2 signaling in the CR mice, they wanted to look at the benefits of this, like why was this good, and they wanted to look at if it was dependent solely upon type 2 signaling. All the effects that we've seen with the brown tissue so far were dependent upon that. So they looked at cell density and tissue volume. So they're basically looking at the browning of tissue and weight loss. In figure D, they had 2 groups of mice. They had IL-4 mice, or they had 4 groups of mice. They had IL-4 mice, negative, so the gene was knocked out in a calorie-restricted group and in a regular diet group. And then they had basically just a control.
And what is IL-4?
IL-4 is the type 2 cytokine. And so in IL-4, when the gene was not knocked out, they had a regular diet group and a calorie-restricted group. So in the top row of figure D, you can see that in the AL group, the tissue was not very dense. And then in the calorie-restricted group, the tissue is obviously a lot more dense, and the tissue did go through browning. However, when they knocked out IL-4, and they knocked out type 2 immune signaling, the benefits of the browning of tissue, they were completely negated. The tissues looked exactly the same, and the calorie-restricted mice did not go through that browning. And then in figure N, they also knocked out IL-4, and they also, in these two regions, they're looking at the ING sat and the PG vat, and they noticed that tissue volume did not change from AL group, from the regular diet group, to the calorie-restricted group. So the weight loss benefits of restricting calories by 40% was completely negated by simply knocking out the type 2 immune signaling. So then, the last thing they looked at was if the type 2 immune signaling would negate the benefits of the browning of tissue, the metabolic benefits. So again, they had 4 groups of mice. They had IL-4, they had the gene, and then there was a calorie-restricted group and a regular diet group. And then they had a knocked out IL-4 group for calorie restriction and for a regular diet. So figure C, we had the results that we've already seen before. They injected them with, or they gave them blood sugar, or they gave them sugar. And their blood glucose levels in the CR mice significantly went down faster than the regular diet group, which we've already established. But then in D, when they knocked out the type 2 immune signaling, the blood glucose levels basically were identical for the calorie-restricted group and the regular diet group. So by simply knocking out that one protein in the type 2 immune signaling pathway, the metabolic benefits of that brown tissue were completely negated. So they found the missing link. They found that calorie restriction led to an upregulation in type 2 signaling, which led to brown tissue and then metabolic benefits from that brown tissue and the weight loss associated with the calorie restriction. And they also found that it is solely dependent on type 2 immune signaling. Without type 2 immune signaling, we get none of the benefits from the calorie restriction. So in conclusion, calorie restriction leads to browning of tissue, which provides its metabolic benefits, such as oxygen consumption rate and the processing of blood glucose. And the effects of calorie-restricted diets are solely dependent upon type 2 immune signaling. Without it, nothing happens, nothing works. And then, now that we've established that calorie restriction and weight loss because of calorie restriction is dependent upon type 2 immune signaling, this is an avenue possibly of further research, maybe even develop a weight loss drug one day. Any questions?
Yeah, a couple slides back you had a gene, it was like the OB gene, and was that something that they identified in a past study, or like what led them to use that gene in the procedure?
The OB gene is basically just like, they had mice and they made sure they had the OB gene. The OB gene basically just makes the mice obese. Basically they're genetically doomed to become obese.
Here's identified wild. If you can Google pictures of baby mice, you just say I'm recommended. I have a couple of questions. Okay, so do you know how calorie restricted in this level in the study?
Yeah, they restricted them 40%. So
Oh,
they're eating
40%.
So the CR group is eating 60% of what the regular group was.
Okay. And then did they this is probably more of an introduction discussion kind of question, but do they have any guesses why restriction of calories would lead to this increased immune response?
No, they didn't really talk about it. I mean, they just found that it was reliant upon that. That could also be an avenue for further research, like why restricting those calories did lead to an immune response. I mean,
There's like a lot of, I forgot to remember its name in the discussion. There's a lot of stuff like about set point theory going on right now, and how like basically your body has a set threshold that your weight like wants to stay in to stay healthy. And if you exceed that threshold in either direction, your body will adapt to keep you within that range. So maybe the immune response is like part of that to help you maintain your natural body's weight, and then once you trigger that, you're able to lose down. I don't know, just a thought.
Yeah, I also saw something about like, what's that show, The Biggest Loser, and like people lose absurd amounts of weight, and then they found 6 years following that they pretty much the person had regained a lot of their weight, and in doing that like highly, losing weight so quickly, they found that for their weight 6 years later, their metabolic rate had slowed by like P80. I don't know what that means, but like it's just interesting.
Do you know if the like changing of tissue to brown tissue, does that like last permanently or does it like revert back to
I'm actually not sure. That's a good question. I get back to it.
Is a brown tissue like located in specific spots or like white fat?
Well, yeah, they were focusing here on the visceral fat, which is like covering your organs, and then the ing sat, I forget what it's called, but it's the subcutaneous fat, which is right under the skin. So that's like where they were focusing that on. But anywhere where you have white tissue, that can be converted to brown tissue.
Did they look all at the, I guess, like the rate of eating? So not like how fast you're eating, but how often? Like, did they look at intermittent fasting at all?
The only thing that they really controlled was just like how much they ate per day. Not really like how often they ate or whatever. Just the total calories they're getting per day. Thank you. Oh.